Welcome to Bosker Sciences, and today's topic is on cell communication. There are 5 main objectives that are going to be covered in today's video, which are going to be quorum sensing, direct contact, local versus long-distance signaling, stages of cell signaling, and lastly, apoptosis. Bacterial cells are able to communicate through a phenomenon called quorum sensing. This process allows bacteria to monitor their population density and use that information to control gene expression. Universal quorum sensing languages even enable bacteria of different species to talk to each other. Many bacterial behaviors are regulated by quorum sensing, including gene transfers and harmless bacteria transforming into a dangerous virulent one. How do bacteria know when their population is large enough? They release signaling molecules into their environment. As the bacteria population increases, so does the concentration of signaling molecules until there are enough molecules to bind to receptors inside or on the surface of all the bacteria, and they trigger a cascade of chemical reactions. These reactions alter the expression of certain genes in the bacteria, turning on the ones that code for enzymes like luciferase, which trigger the expression of the protein luciferin and produce light. Take a look at the diagram. Bacterial genes code for the production of signaling molecules called autoinducers that are released into the surrounding environment. These signaling molecules then bind to the signaling receptors either on the bacterial surface or in the cytoplasm, in this case, on the surface. When these autoinducers reach a critical threshold level, they activate bacterial quorum sensing genes that enable the bacteria to behave as a multicellular population rather than as individual single-celled organisms. The autoinducer receptor complex is able to bind to DNA promoters and activate the transcription of quorum-sensing controlled genes in the bacterium. In this way, individual bacteria within a group are able to benefit from the activity of the single group. One type of signaling is called direct contact, and that's where cells communicate with each other. Signaling substances dissolved in the cytosol can pass freely between adjacent cells. Gap junctions permit the passage of materials directly from the cytoplasm of one cell to the cytoplasm of another cell. In muscle tissue of the heart, the flow of ions through gap junctions coordinates the contractions of cardiac cells, and plasmodesmata connects one plant cell to the other. They are basically the same as gap junctions in animal cells. There are 2 more types of signaling, and they are local and long-distance signaling. The release of local signals, such as growth factors, from one cell to nearby cells, give instructions to grow and divide. An example of this is synaptic signaling, which is a neuron releasing a neurotransmitter into a synapse to stimulate an adjacent neuron to fire or a muscle to contract. Plants, however, use chemicals called hormones for long-distance signaling. In animals, the endocrine system has endocrine glands to release hormones in the blood to reach target organs or structures. Another example of this is how helper T white blood cells send out an alarm to the entire immune system that the body is being invaded by bacteria or viruses. If you look at this diagram, you can see that there's 2 types of local signaling, which are paracrine signaling and synaptic signaling. Paracrine signaling is a secreting cell that secretes signaling molecules into the extracellular fluid when target cell is nearby. Meanwhile, synaptic signaling is when a nerve cell releases neurotransmitter molecules and that diffuses across the synapse, which is the space between a nerve cell, and that gives a chemical signal to a new cell. On the right of the diagram, you can see that it is long-distance signaling, aka endocrine signaling, and hormones travel through the circulatory system to target cells, and hormones are used for long-distance signaling. We talked about the different types of cell signaling, but now we're going to move more into the steps of cell signaling. The first step is reception, and that's when a ligand from the outside cell binds with the receptor in the target cell. The second step is transduction, and that's when a signal is converted into information that can be used by the cell. The last step is response, and that's a specific cellular response either by transcription or cytoplasmic action. The first type of receptors that we're going to talk about are the ones that are on the cell surface. What you should know is that all cell membrane receptors are similar because they span the entire membrane and are therefore in contact with both the extracellular environment and the cytoplasm. Hydrophilic signaling molecules can't diffuse through the membrane. However, they bind to the part of the receptor on the cell surface to change the shape of the cytoplasmic side of the receptor. Once it's inside the cell, the signal is carried by a second messenger. The most common second messenger is cyclic AMP, also known as CAMP. Notice that the first messenger, or the ligand, never enters the cell. There are three main types of cell surface receptors, and they are ion channel receptors, G-protein coupled receptors, and protein kinase receptors. Ion channel receptors are allosteric receptors that open and shut a gate in a membrane, allowing an influx of ions such as sodium, potassium, or chlorine. These ion channel receptors and ion-gated channels are the basis of normal nerve function. G-protein coupled receptors span the entire cell membrane. When a ligand binds to the extracellular domain of the receptor, it changes the conformation of the cytoplasmic side of the receptor. This activates the G protein, which is an on and off molecular switch, and then when the G protein is activated, it bonds to the GTP, which is a high energy nucleotide that acts like ATP, and that activates the enzyme adenylyl cyclase. This enzyme catalyzes the conversion of ATP to CAMP, the second messenger. It is this CAMP that activates the other molecules inside a cell, leading to a cellular response. The next type of cell surface receptors are receptor tyrosine kinases, and after binding, the units aggregate and activate the kinases which bond to ATP. The next type of receptors are intracellular. Small nonpolar ligands diffuse directly through the plasma membrane and bind to an intracellular receptor in the cytoplasm. Such hydrophobic chemical messengers include steroids, thyroid hormones, and nitrous oxide, and they all readily diffuse into the cell and switch an on or off gene in the nucleus. An example is estrogen, which diffuses through the plasma membrane, binds to a receptor in the cytoplasm, and initiates a response inside the cell. A chemical that you should know is bisphenol A, and that is a chemical present in many plastics which mimics hormones like estrogen. Bisphenol A is known as an estrogen mimic because it sets up an estrogen signal transduction pathway. It is believed to be responsible for problems in fetal development in humans. Once activated, the receptor converts or transduces a molecular signal into a cell response. This transduction sometimes occurs in a single step, but more often it requires a sequence of changes in a series of different molecules, which is also called a signal transduction pathway. A signaling transduction pathway is a multi-step process in which a small number of extracellular signaling molecules produce a cascade effect. The advantage of this complex set of reactions is that the multi-step pathway produces more opportunities to amplify the signal greatly. Signal transduction pathways exist in both yeast and animal cells, and in bacteria and plants. The strikingly similarity in these pathways among diverse organisms suggests that the pathway evolved hundreds of millions of years ago in a common ancestor. You don't have to memorize this, but I just wanted to show the signaling transduction pathway of insulin in the human body. The last step of cell communication is response, and that's when a signal transduction pathway leads to a response which turns genes on or off. And there are 5 things that you should know about signal transduction pathways. Number 1 is that they're characterized by a signal, transduction, and response, which we already talked about. Number 2, they are highly specific, so they only occur with one type of cell or one type of receptor. Number 3 is that one signal molecule can cause a cascade effect that can cause many molecules to form. Number 4, it also regulates cellular activity. Number 5, they evolved millions of years ago from a common ancestor. The last topic that we're going to talk about is apoptosis, and that is a genetically programmed series of events that results in cell death. During this process, the DNA, organelles, and other cytoplasmic components are chopped up. The parts are packaged in vesicles that are engulfed by special scavenger cells. Apoptosis relies on several different cell signaling pathways and the signals from outside, and sometimes from inside the cells. There are 4 main reasons why cells carry out apoptosis. Number 1 is for embryonic development, and an example is that the tail of the tadpole, which undergoes apoptosis during frog metamorphosis. Number 2 is that the cell has sustained too much genetic damage that could lead to cancer. This is common for epithelial cells on the surface of the skin, which have been exposed to extensive solar radiation. Skin cells normally die by apoptosis and are replaced every few days, but sometimes there's genetic damage like we talked about. The third reason is to defend against fungus and bacteria, and the last reason is to erase protein misfolding in cells. So that wraps up today's video, and I hope you like the video, and if you did, make sure you subscribe and like it.